Hello, I'm Jan Leifman.
I'm Gayatri Menon.
Welcome to Med News Week Conference, where we feature presentations by medicine's global leaders. Today, we have an amazing keynote speaker in Dr. Seth Pollack. Dr. Pollack is the director of the Sarcoma Program at the Lurie Cancer Center of Northwestern University.
In today's presentation, Dr. Pollack discusses the latest in sarcoma therapeutics.
Did you know that Dr. Pollack is an internationally recognized leader, physician, researcher, and speaker on sarcomas? He has served as the chair of the National Leiomyosarcoma Foundation Grant Review Committee and received numerous national awards, including the SIG Kohl Legacy Award, the Northwest Sarcoma Foundation, the V Foundation Translational Research Award, and the Sarcoma Alliance for Research and Collaboration Career Development Award.
He's truly a dedicated advocate and a leader in the field of oncology and clinical trials.
So let's tune in. Let's tune in and learn from this great global leader.
Sarcomas are a type of cancer. But they're not just one type of cancer. There's a lot of different types of sarcoma. The sarcomas comprise about 1% of all cancers, which sometimes people will poo-poo. They'll say, oh, that's not important. But actually, 1% of all cancers is a lot of people. So many people are affected by cancer. And in the US, 1% of all cancer is over 20,000 people a year. So that's a fair amount of patients. And we divide that up into the bone sarcomas. So you may have learned about osteosarcoma, Ewing sarcoma. Those are two sarcoma types that tend to affect kids and young adults. Those are both very sensitive to chemotherapy, and chemotherapy is a really critical part of the treatment paradigm for those diseases. Chondrosarcoma is another very important type of bone sarcoma. And then there's the soft tissue sarcoma, and what used to be just another type of soft tissue sarcoma only 20 years ago, today GIST is really very different in terms of its treatment paradigm. And we know that in most cases, GIST is driven by mutations in the KIT gene. And because of that, it can be very sensitive to imatinib, sunitinib, regorafenib, ripretinib. And now there are new agents that are very specifically targeting specific mutations, such as avapritinib, which specifically targets the exon 18 PGFR alpha mutations that can occur in GIST. Rhabdomyosarcoma are a group of sarcomas, and frequently in kids, they'll be embryonal or alveolar rhabdomyosarcoma, which have their own chemotherapy treatment paradigm. But today, I'm going to be focusing on the rest of the soft tissue sarcomas. Over 13,000 in the US patients per year, most sarcomas, which are soft tissue sarcomas, which is still a great variety of tumors, as we'll discuss. There's so many different types of sarcoma, it can be overwhelming. And this is from a paper that was focused on the ultra rare sarcomas. So you can see that a majority of soft tissue sarcomas have one of the most common subtypes. So undifferentiated pleomorphic sarcoma, a lot of us back when we were in school, we used to call those MFH, or malignant fibrous histiocytoma. Today, the nomenclature is different, and a majority of those old, very poorly differentiated sarcomas are called undifferentiated pleomorphic sarcomas. But there's a lot of little flavors within that, spindle cell sarcomas that can be poorly differentiated, and a lot of different types. Liposarcoma, another generally common type of sarcoma, and there's a few types of liposarcoma. Well-stroke dedifferentiated liposarcoma, which are driven by MDM2 and CDK4. Myxoid round cell liposarcoma, which we're going to talk about quite a bit, which is a translocation-driven sarcoma. Pleomorphic liposarcoma, which is a different type of liposarcoma. Leiomyosarcoma is another common type of sarcoma. These are cancers of the smooth muscle, and most people think, well, if it's a cancer of the smooth muscle, it must come from the small intestine, right? That's where so much smooth muscle is, but that's not the case, right? So about a third of leiomyosarcomas are uterine, and then Many of the other leiomyosarcomas originate in the smooth muscle that surround vascular walls, and especially in the retroperitoneum, in the smooth muscle surrounding some of the great vasculature. Like, people can develop leiomyosarcoma from the IVC, or from, it doesn't have to be from the IVC, it could be from smaller blood vessels. People can also get leiomyosarcoma in their skin or in their extremities in subcutaneous tissues. We'll also talk a bit today about synovial sarcoma, which is about 8% of sarcomas. It's another translocation-driven sarcoma. So you can see there's a lot of variety of sarcoma. These different tumors can be very different from one another. I mean, if you just imagine the difference between a liposarcoma and osteosarcoma, which we were just talking today, that's the difference between a cancer of the bone and the cancer of the fat. And they have very different biologies, very different treatment paradigms. But today I'm going to be focusing about the immunobiology of these tumors, which, as you'll see, varies quite a bit, just like their treatment paradigms do. And we're going to be talking today about cold tumors and hot tumors, which I think is a very reductive model of how we think of the immune system in cancer. But I think sometimes it can be helpful to think about things in this way. So cold tumors, of course, I know that you guys have had a lot of great speakers, some of the world's experts in immunotherapy, who I'm sure have brought up this concept before, but I'll just review that cold tumors tend to not have a lot of T cells. They frequently have a lot of immunosuppressing cells that could be like macrophages, which can have an M2 or immune inhibitory phenotype. They could have other immune suppressing cells like T regulatory cells, etc. They often have low expression of antigen presenting molecules like MHC. And sometimes they can have high levels of inhibitory cytokines that are just in the tumor. And this contrasts with hot tumors, which have a lot of T cells that tend to be exhausted in the tumor. And those tumors frequently will have high levels of PD-L1. They will also have immunosuppressive myeloid cells like macrophages or myeloid-derived suppressor cells. Those tumors actually are more likely to have regulatory T cells. The regulatory T cells are probably a bigger component of the inflamed tumors on average. So on the one hand, having, it used to be that, now we live in the age of immunotherapy. So it's easier to fight a hot tumor with a PD-1 inhibitor or CTLA-4 inhibitor or any of the checkpoint inhibitors, because those tumors already have exhausted T cells that are in the tumor. Presumably, those T cells are specific for the tumor. And if we could just figure out a way to turn them on, then they would kill the tumor. So, in theory, when you're talking about immunotherapy, these hot tumors are closer to being targets for immunotherapy than the cold tumors. I'm sorry. So the three kind of things I want to talk about today is our efforts to define what a normal sarcoma immune microenvironment is. And then we'll talk about the problem of inhibitory macrophages, which is what we think the crux of the problem is for sarcoma immunotherapy. And then we'll talk about how we can change the microenvironment to facilitate cellular therapy in sarcoma. So what's a normal sarcoma microenvironment? And we'll also be talking a little bit about radiation and how that impacts it, because that's a normal part of sarcoma treatment. And we did a study to try and characterize the normal sarcoma microenvironment, and we looked at some of the most common sarcoma subtypes: the undifferentiated pleomorphic sarcomas, the liposarcomas, the leiomyosarcomas, and we looked at synovial sarcoma because that's it's a very as you'll see later, it's a hot topic for immunotherapy, and this included two sarcoma types that have complex genetics, frequently the undifferentiated pleomorphic sarcoma and leiomyosarcomas, and two with relatively simple genomes: the synovial sarcomas, which are translocation driven, and liposarcomas, which can either be dedifferentiated liposarcomas, which are driven by MDM2 and CDK4, or the myxoid round cell liposarcomas, which are translocation driven. And you can see, although this figure out is, I don't know why it's not coming out so clearly, but the undifferentiated pleomorphic sarcomas have significantly higher levels of PD-1 and PD-L1 by immunohistochemistry. So this was very clear from the start. There are some patients with synovial sarcoma that will have PD-1 or have intermediate PD-L1, and those tumors don't respond to PD-1 inhibitors. Even leiomyosarcoma, which does not respond to PD-1 inhibitors, can have some PD-1 or PD-L1 staining, but it's the undifferentiated pleomorphic sarcomas that unquestionably have higher levels of PD-L1 expression in the tumor. And we looked by nanostrain to characterize the immune-related gene expression in these tumors. And what we found is there was a striking separation among these tumors for genes related to antigen presentation and T-cell infiltration. So, of course, because I know there's all sorts of folks in this very large audience that you guys have, just a reminder that when a T cell, like a CD8 T cell, sees a tumor, they really need to see antigen, and they don't just see antigen on the cell surface, they see antigen presented by MHC class 1, right? So, we were interested in characterizing that antigen, that antigen presentation, which we saw was different between these tumor types. And you can see that the two translocation-driven sarcomas, synovial sarcoma and mixed red round cell liposarcoma, have clearly less expression of the CD3D, which was we used as a marker of T cell infiltration, and clearly less HLA-A, which is HLA and MHC are the same. HLA-A is one of the MHC related molecules, right? So they had significantly less, and the undifferentiated pleomorphic sarcomas had more when it comes to antigen presentation, and both the undifferentiated pleomorphic sarcomas and leiomyosarcomas on average had more T cells and more antigen presentation. And this is a bit tricky because the leiomyosarcomas don't seem to respond to checkpoint inhibitors just generally. They have a wide variety of expression of these things. But the undifferentiated pleomorphic sarcomas do, as we'll talk about in a minute. IL-7 receptor was also a marker for T cell infiltration, which was lower in the synovial sarcoma and mixed red round cell liposarcomas. And the same was true for HLA-DP, which is a class 2 MHC or HLA. So that this is kind of the tumors don't generally express class 2 MHC, but could be reflective of antigen presenting cells in the tumor microenvironment, myeloid cells, immune cells in the microenvironment. We also did TCR sequencing, and I'm going to talk about TCR sequencing quite a bit in this talk. So remember back to your immunology days, when T cells differentiate, or when T cells form, they during development, they undergo rearrangement of their T cell receptor, right? That's how you get such a incredible diversity of different T cells, right? Your T cell population in your body, you have all the T cells that you need right now to recognize whatever is going to come your way, be it cancer or infection. And the thymus, of course, during development will help to eliminate the autoreactive T cells. But because these T cells have undergone a gene rearrangement, you can do a sequencing to describe the T cell repertoire. So you can see over here, this is one of our samples. So on your lower left-hand corner, this is a TCR sequencing done on the Adaptive Biotechnologies platform. And we sequenced one sample, and you can see this has a number of different, it has lots of different TCRs in there, T cell receptors, TCRs. And the most common TCR that was in this sample comprised 0.27% of that sample. And you can see the amino acid sequence. And then the next one was 0.15, and you can see the amino acid for that one. And you can summarize this by a metric called clonality. And I've taken this graph in the middle here is a graph to illustrate the concept of clonality. It actually means something, and we're going to discuss it later on. But here, I'm just going to use it to talk about the concept of clonality. So you can see here that in the dark red, this is an illustration of how much each T cell receptor is comprising the total one. So you can see the dark red is comprising the most common T cell receptor that was in this sample, and that makes up about 10% of the T cell receptors in a sample. By contrast, the top 10% over here is over 9,000 different T cell receptors. So that's the top of here. Now we did a treatment, and we'll talk about what the treatment was, and then this sample changed. So now the top TCR comprised almost 40% of the T cell receptors in the sample, and the TCR is 1,001 to 10,000, so 9,000 TCRs. The 9,000 lesser common TCRs comprised less than only a few percent of this sample. So this would be a sample where we would say the clonality is very high because it's skewed towards the most common T cell receptors. So we did this with our samples, and first of all, we characterized by using this technique, you can actually be very accurate in terms of quantitating the number of T cells that are in a sample. And you could see that the synovial sarcomas had a very low T cell fraction. The myxoid round cell liposarcomas also had a very low T cell fraction. And the undifferentiated pleomorphic sarcomas had a significantly higher T cell fraction. And this was true of the clonality as well. So the synovial sarcomas tended to have a low clonality, as did the myxoid round cell liposarcomas, as did the other liposarcomas. But the leiomyosarcomas had a little bit more, and the undifferentiated pleomorphic sarcomas had a significantly higher clonality. And what we think this means is we think this is reflective of the function of the T cells in there, that if your clonality is low, that it's more likely that the T cells are just randomly assorted there because there's no specific clones that have seen their antigen, have started to replicate, and have become a more dominant portion of the T cell repertoire in those tumors. Whereas the ones with a high clonality, there are some dominant clones that are probably specific for tumor antigens. And these results with regards to the tumor histologies are, at least with respect to undifferentiated pleomorphic sarcoma, it seems to be consistent to what we see in terms of response to PD-1 targeted therapy. So you could see here undifferentiated pleomorphic sarcoma in this landmark study, the SARC-28 study. 4 of the 8 patients treated with pembrolizumab here had durable responses to this pembrolizumab, a PD-1 inhibitor. And the dedifferentiated liposarcoma, which is somewhere in the middle, they had some patients with disease stability, maybe some responses. I mean, they did see 2 responses, but they were not as deep or durable as the undifferentiated pleomorphic sarcomas, but they had some durable disease stability. But no responses with leiomyosarcoma, 1 kind of very short-lived response in synovial sarcoma. I'm just going to emphasize the point right now that liposarcoma is 2 different diseases, right? The well-stroke dedifferentiated liposarcoma, and well, it's 3 diseases, but I'm going to be talking about 2, the 2 most common ones, right? Pleomorphic liposarcoma is an unusual one. There are people who are saying maybe there's more than 3 types, but I think there's kind of 3 types that everybody agrees with. And when I talk about well-stroke dedifferentiated liposarcoma, some people just have well-differentiated liposarcoma, and that generally has a good prognosis. The histology looks like normal fat, but if you have dedifferentiated disease, this is a very different tumor type. It's a very different tumor appearance histologically. So this patient has both a well-differentiated liposarcoma tumor, that's this dark one over here, and a dedifferentiated tumor over here, that's this lighter tumor. And the dedifferentiated liposarcomas, that's more of the aggressive portion. That's the portion that's going to really cause the patient problems. Myxoid round cell liposarcoma is called that because there's two components to it. There's a round cell component, which on this histology section is at the bottom, and the myxoid component, which is at the top. And if you're just seeing a patient for the first time, the round cell component is the component of the tumor that is conveys a worse prognosis that's associated with an aggressive clinical behavior. And you need to really watch out if your myxoid round cell liposarcoma patient has a big round cell component in their tumors. And we took a really careful look at the immune microenvironments in these liposarcomas, and we found that there were certain immune characteristics that were associated with a favorable outcome, with a recurrence-free survival that was over 3 years. And the big one was the presence of B cells. CD4 cells also made a difference. And then actually, the big one against this good survival was the macrophages and CD4 cells, or the macrophages and CD14 positive monocytes. CD4 cells were pushing things in the right direction. And we did this TCR sequencing for these dedifferentiated liposarcomas as well. And we found something really surprising, and it is something I don't know how to explain, something that my lab is really focused on right now. It's something that I think is a really fascinating question, is that when you looked at, so this is a chart where you map all of the different samples with respect to their T cell fraction, what percentage of the cells in the sample were T cells, and the T cell clonality, which is what we were just talking about before. And you could see that if you were in the upper left quadrant, we would think that probably that wouldn't be such a bad thing, because as I said before, a high clonality represents what we think is an antigen-specific response. But if you were in the upper left quadrant, it had a huge impact on your survival. So you could see that not in the upper left quadrant, lots of patients were living for many, many years after their resections, whereas not so if you had a T cell receptor profile that was in the upper left quadrant here. That's something that I don't understand, and something that we're trying to sort out now. What about the macrophages? As I told you at the beginning, we think the macrophages are such a critical part of the tumor immune microenvironment in sarcomas. And you can see that this is, just because I know that not everybody does immunology all the time, just a reminder that macrophages have a lot of different functions, and they are an immune cell that is in charge, that plays a role in defending the host immunity, right? So they can present antigen. They can produce inflammatory cytokines like interferon gamma and TNF alpha, and they present antigen to T cells to stimulate the T cell response. And there are markers like TLRs, like CD8086, like MHC class 2, like INOS, that signal that this is an inflammatory macrophage. This is a macrophage that's there to improve immunity. Likewise, macrophages also play an important role in wound healing. And in the midst of a wound, it doesn't help anybody to have a big inflammatory response when you're trying to heal somewhere. And that's why, those macrophages are producing cytokines like IL-10 and that tend to be NTGF-beta, that tend to be immune inhibitory and prevent the T cell response. And those macrophages tend to express CD163, CD206, et cetera. So those are M2 or inhibitory macrophages. And we know that these macrophages are in a lot of different types of sarcoma. This was a great publication that looked at a lot of different types of sarcoma, characterized the macrophage infiltration patterns. You can see that most of these sarcoma types have a lot of macrophages, but especially in all the more common types of sarcoma that we've been talking about, the undifferentiated pleomorphic sarcomas, the liposarcomas, the leiomyosarcomas. Just a point that the leiomyosarcomas have a fair number of this CD163 positive macrophages. And we saw this in our data sets as well. Lots of different similar macrophages across all the different types that we looked at, but probably a little bit more in the liposarcomas, the undifferentiated pleomorphic sarcomas. And we looked at the immune infiltration in myxoid round cell liposarcomas and synovial sarcomas in particular, because I'll be talking about later, we're really interested in targeting these with immunotherapy. These are very immunologically quiet tumors. We've already mentioned that they have relatively low numbers of T cells. They have relatively low numbers of immune cells across the board, but they have a lot of macrophages relative to these other cells. And in leiomyosarcoma, it's been really well documented that if you have inhibitory macrophages, that they're especially if you could see these two graphs at the bottom. This was seen not in uterine leiomyosarcoma, but in non-uterine leiomyosarcoma, there was a statistically significant worsening of outcomes when a patient had inhibitory macrophages that were seen on IHC. So one thing that we know, so we were really interested also in how radiation impacts this. And I'm going to come back to macrophages in a second, but we looked at patients with sarcoma getting neoadjuvant therapy, because a lot of these patients will get radiation. Most of these patients also had chemotherapy too, but we just as part of our standard clinical practice, we would collect a biopsy, then a patient would get neoadjuvant therapy, which almost always included radiation, and did include radiation here, sometimes included some chemotherapy as well, and then they would go for surgery. So what we did is we compared the immune microenvironment from the patients before their neoadjuvant therapy to after their neoadjuvant therapy. And you can see we had a whole different variety of different sarcoma types, a diversity in sarcoma sizes, grade, mainly intermediate or high grade, but a lot of different types of sarcomas. And what we saw is a big increase in the antigen presentation molecules like HLA-DR. We saw a big increase in B cells and natural killer cells, and we saw an upregulation of genes related to antigen presentation. And we saw a big increase in macrophage-related markers. So on the one hand, while radiation might be killing the tumor and thus is absolutely essential for improving outcomes of these patients, we know that that's incontrovertible with excellent data. We may also be increasing the inhibitory macrophage population in these tumors with radiation. We thought that maybe radiation would be sensitizing these tumors by just increasing the T cell infiltration and increasing the antigen presentation, which it does, but it also has a big impact on the M2 macrophages, as you can see by the increase in CD163, the increase in CD206, etc.